Welcome to the Bioinsights, where science comes alive.
You created antibiotics to kill bacteria, but I learned how to survive them.
I am a drug-resistant bacterium, a superbug shaped by evolution, mutation, and antibiotic selection. When antibiotics eliminate my susceptible neighbors, the survivors can multiply, and resistance can spread. Antimicrobial resistance is one of the major challenges facing modern medicine, increasing the need for new antibacterial strategies.
Traditionally, discovering antibiotics means screening enormous numbers of compounds, testing them repeatedly, and eliminating candidates that fail.
The chemical universe is enormous. Testing every possible molecule experimentally is simply impossible.
So humans gave me a challenge. Search the chemical universe faster than they ever could.
I can learn patterns from chemical structures, biological activity, bacterial targets, toxicity data, and enormous scientific datasets. To me, a molecule isn't just a picture. I can represent its atoms, bonds, properties, and relationships mathematically. Instead of testing everything, I can prioritize molecules that are predicted to have antibacterial activity. And sometimes I can identify structures that human researchers might never have selected using conventional approaches. One famous example is halicin, identified through a machine learning approach and shown to have broad antibacterial activity. Another example is abaucin, identified using machine learning for activity against the difficult pathogen Acinetobacter baumannii. You found new molecules? Interesting. But remember, I'm an evolutionary expert. Whenever pressure changes, bacterial populations can evolve. Your next antibiotic may eventually face resistance too. I can also analyze genomic and biological data to help researchers predict resistance patterns and understand bacterial vulnerabilities. Before designing an antibiotic, researchers need to understand where and how it might attack the bacterium. Computational models can help predict how candidate molecules might interact with bacterial proteins.
A strong computational prediction doesn't automatically make a good medicine.
A potential antibiotic also needs suitable pharmacokinetics, safety, stability, and toxicity characteristics.
And now comes something even more exciting. I can help generate entirely new molecular structures.
This approach is called de novo molecular design, creating candidate molecules rather than simply searching existing libraries.
In minutes, AI can propose enormous numbers of possibilities, but only a small fraction may survive scientific testing.
This is where biology takes over. Predictions must leave the computer and enter the laboratory.
We test whether the candidate actually inhibits bacterial growth under controlled experimental conditions.
Then we investigate its potency, mechanism, selectivity, and potential toxicity.
An antibiotic doesn't need to kill every bacterium. Sometimes a narrow-spectrum molecule targeting one dangerous pathogen can be extremely valuable.
Targeted antibiotics may offer an important advantage by focusing treatment on specific pathogens rather than unnecessarily disturbing beneficial microbes.
But I don't replace microbiologists. I help them decide which questions and molecules deserve closer investigation.
AI is powerful, but it can learn from incomplete data, produce false predictions, and struggle when real biology behaves differently from its training data.
Most promising computational candidates will not become medicines. Drug discovery is a long journey filled with failure. Even a successful laboratory candidate must pass many stages before it can become a medicine for patients. Only rigorous clinical research can establish whether a new antibiotic is safe and effective in people.
Even if you discover your next weapon, misuse it, and I may evolve again.
New antibiotics alone won't solve resistance. Responsible prescribing, infection prevention, surveillance, and stewardship remain essential.
The future may belong to a partnership between artificial intelligence, microbiology, chemistry, genomics, and experimental science. I can search faster. Scientists can test smarter. Together, we may explore chemical space that was once far beyond our reach. So, can AI discover the next antibiotic? The answer is potentially yes, but discovery is only the beginning.
The real battle isn't AI versus bacteria. It's humans using science intelligently before resistance leaves us with fewer options.
The next antibiotic may begin as a line of code, a molecular prediction, or an unexpected idea.
Follow the BioInsights as we explore the technologies fighting the world's most dangerous microbes. Subscribe and discover where biology meets the future.
Thanks for watching the BioInsights. Like, subscribe, and follow for more.